The overall goal of this method is to isolate viable mucosal myofibroblasts and fibroblasts from frozen gastrointestinal tissue samples originally obtained from biopsies or surgical specimens.
The isolation of myofibroblasts and fibroblasts from properly frozen gastrointestinal tissue allows processing of fresh samples to be delayed. This facilitates the use of samples from different sources worldwide.
If the tissue was preserved in freeze medium, place the cryovial in a 37 degrees Celsius water bath until the tissue and medium are thawed. For 4 to 5 2 square millimeter tissue fragments, add 10 milliliters of HBSS without calcium and magnesium. Tightly close the lid and wash the tissue by gently inverting the tube one time. Once the tissue has settled by gravity, carefully discard the supernatant and wash the tissues again. Then add 10 milliliters of collagenase solution to the tube and transfer the tissue suspension into a sterile DNase-RNase-free tube with built-in rotors. Tightly close the rotor tube and attach it upside down onto the sleeve of a dissociator. Then run the preset HTumor01 program. At the termination of the program, incubate the tissue slurry for 45 minutes at 37 degrees Celsius under continuous 140 RPM rotation on a shaker. Then return the tube to the dissociator and run the HTumor02 program.